Hello, my name is Michael, and today I'll be talking about improving chemotherapy against acute myeloid leukemia. I'll be doing this by looking at the study targeting cell cycle and apoptosis to overcome chemotherapy resistance in acute myeloid leukemia. So to understand how chemotherapy works against acute myeloid leukemia, or AML, we first need to understand what AML is. Well, it is a type of cancer that affects blood and bone marrow. This is caused by immature precursor cells called myeloid blasts, and these myeloid blasts cause crowding of the bone marrow, leading to anemia, which is from low red blood cells, infection, which is from low white blood cells, and bleeding and bruising, which is from low platelets. Furthermore, 1 in 5 people also develop cancer in their lifetime, and 1% of those people will develop AML, meaning 0.2% of the whole world will get AML at some point. And so why focus on chemotherapy resistance against AML? Well, that is because 15% of people who undergo chemotherapy fail and don't respond at all to therapy. And with the 85% of people who do respond, half of them relapse, meaning treatment for them failed too, leaving only 42.5 people with successful treatment and 67.5 of people with unsuccessful treatment. To understand what makes AML so difficult to treat, we first need to understand how AML cells disrupt the processes that regulate its growth. One of these processes is the cell cycle. The cell cycle allows the cell to grow and multiply by going through checkpoints. These checkpoints are the G1 phase, where the cells begin to grow, the S phase, where it synthesizes DNA, G2 phase, where it grows and prepares for mitosis, and the M phase, where mitosis occurs and cell division occurs, creating two identical daughter cells. Another process is apoptosis, which just programs cell death. This programmed cell death is meant to prevent cancer cells from proliferating. So, what is chemotherapy? Well, it is simply a combination of drugs called cytarabine and doxorubicin. These drugs interfere with cancer cells' ability to avoid regulation and cell death. And now that we understand how AML cells generally avoid regulation, we now need to understand the concepts used in the study. One of these concepts is CRISPR-Cas9, which is a genetic tool that is used by bacteria in order to fight off viruses. It is used as a way to manipulate specific genes in the sequence. And it does this by matching specific sequences we want using gRNA. This gRNA then recruits a protein called Cas9 to the location we desire. This Cas9 protein has the ability to cut off the gene, causing it to turn off. It is also called knockout. Another concept is called Brunello gRNA library, which is a CRISPR-based tool that is used for genome-wide knockout. This library contains a gRNA match for every gene that encodes for every protein in the genome. And the last concept is called next-generation sequencing, which just uses a computer to sequence the entire DNA and RNA quickly and accurately. In order to find out which genes were important in chemotherapy resistance, they created two tests, one in vitro, where they focus on identifying key genes, and one in vivo, where they focus on creating new chemotherapy combinations. For the in vitro test, they got Cas9 and put it into an AML cell line plate using lentivirus as a vehicle. They did the same thing with gRNA library, where they put it in using lentivirus. After that, they got a culture plate that has AML cells that were able to express Cas9 and gRNA library. These genes then get run through next generation sequencing, where it gets snapshotted. They then introduce chemotherapy into the plate, and then do another next generation sequencing with the cells that manage to stay alive. They then compare the frequency of the genes by comparing it before and after. And if the gene appeared less frequently, it would mean that it was depleted. That means by knocking it out, it caused the cancer cell to die, making it the target of the research because we want to prevent the cell from surviving. But it's also important to know which genes were enriched or appeared more frequently because that would help us understand how genes survive chemo. And for the in vivo test, they just used live mice. Everything was the same, where they snapshotted the beginning, then introduced Cas9 and gRNA library expressing AML cells into the mice. They do that by injecting cells into the tail vein of the mice, where it goes straight into the bloodstream and migrates into the bone marrow, where it develops into leukemia. They then put the mice through chemotherapy, but instead of the same chemotherapy they used in vitro, they mixed and matched with additional drugs, palbociclib and venetoclax. And then after chemotherapy, they extract the cells from where the cancer grew. After that, they put the cells through next generation sequencing and then compare a before and after, similarly to the in vitro test. And what they found when they compared before and after chemo in the in vitro study, they found that these genes are responsible for regulating cell cycle. Meaning if they get knocked out, the cells would lose the ability to regulate cell cycle, causing the cell to proliferate. They also found that these genes were responsible for drug metabolism, which is important because the cell needs to metabolize the chemotherapy drugs in order for the drug to activate and kill the cell. Interestingly, they also found that the enriched genes, or genes that help the cell survive chemotherapy, usually only interfere with the G1 or S phase of the cell cycle. For the in vivo study, the same genes were also enriched and depleted, which validated the results from the in vitro. But interestingly, when they mixed and matched chemotherapy drugs, they found that when palbociclib and venetoclax were used in combination, it was effective in overcoming drug resistance. It was most likely due to palbociclib being able to reinforce cell cycle arrest and venetoclax being able to lower the threshold for apoptosis. In conclusion, by understanding the ways at which AML cancer cells avoid and survive chemotherapy, it allows us to target those specific pathways, which would improve cell cycle control, boost apoptosis, improve drug metabolism. This would increase the effectiveness of current chemotherapy treatments, especially through either generalized or personalized treatments. After considering what they found in the research, I had a few questions, such as how could we identify those with specific genes resistant to chemotherapy? Well, that would definitely be an issue, but you could identify these mutations or biomarkers through genetic screening or by looking at gene expression levels. Another thing that I thought was important was if the patient had primary or a relapse case. Well, since primary cases are more uniform and less variable in terms of genetic population, it would make it easier to treat because treatment for one patient could be used for treatment for another. But for a relapsed patient, it would be different because relapsed AML is usually more resistant. That is how it was relapsed in the first place. So it would require a more individualized and aggressive treatment. And lastly, could mutations of AML eventually catch up to our new therapies? And the answer is yes. AML mutations can evolve in a way where it would make it more resistant to new therapies. That is why it is necessary to have ongoing clinical studies in order to adapt to changes. But on a more positive note, by identifying the key genes involved in cell cycle regulation and apoptosis, it could open more pathways for individualized therapies, such as being able to identify patients who respond best to palbociclib and venetoclax through the use of biomarkers that we could identify. And through even more research, we could discover more combinations in order to discover more AML therapies, which could enhance our ability to treat a wider range of patients who suffer from AML. Thank you.